I am involved in biological farming consulting, and what that means is basically a whole range of different things. So I do soil testing on farm, reading of soil test results, and helping farmers understand what that means, how they can transition into a biological farming future, and also trying to relate results and information of things that we're doing on our farm. Although we're conventionally farming, we are using biological applications here and there and using compost tea perhaps where we can as well. So if you have any questions about that side of it, just ask me, and if I haven't answered it, by all means you can email me or call me after the day and I can get the information to you. So to talk about biological soil applications, the key criteria that we're looking at for success in these sorts of systems is the environment that the biology is going to be put into. So if we're talking about our farms, and I get a lot of people asking me how we're going to go about establishing soil biology onto a farm that's maybe been run down, or over the last 5 months we've had not so much rain, and so how's that going to go about getting established? So we need to look at those sorts of factors that are going to be involved in getting to that stage. One of the key things that we'll be looking at is soil structure and porosity. So we'll do that in our soil testing segment this afternoon. We'll go out and have a bit of a look at that, and also the moisture holding capability, because that is key for establishing soil biological populations. And also the nutrient constraints, so in terms of whether we've got any excesses or deficiencies that are going to hold our progression back in that regard, because it is very much linked, the nutrient side of it is very much linked to the biological side, and magnesium imbalance is one of the things, and also sodium and chlorides, which can be quite common around this area and can impact soil biological populations as well as just general soil health. And the ongoing care, and after we put out the biology, how we're going to foster that to keep growing once we put it out into the paddock. So we'll have a look at that today when we go and do our little walk later on, and we'll talk about the factors that we can influence there in terms of keeping our biological populations growing. The main things we'd be looking at is carbon loads, which is biological food, and also their home, which is also carbon storage, and also the porosity that we talked about earlier. So the spaces, air and water spaces around the soil particle where they're going to live. So who's heard of the soil food web? It's very complex. Yes. So you've probably already read your sheet. So it's a complex web of interactions, and what that means is that we can't isolate one area of the soil food web and just concentrate on that with, forgetting the other aspects of the soil food web. So if we just focus on fungi and developing fungi in our soils, which are really super duper important and which have been neglected for, pretty much forever in our farming systems, starting to change now. But if we just focus on fungi without focusing on the other aspects of the biological system, we're not going to get the best development, because obviously fungi rely on other parts of the system for food and also for predation as well. So turnover of nutrients is directly related to this sort of diagram. So if we have a look at here, what would be the most important thing that we would be considering for our farms? What would be the one thing that we would be looking at first?
Organic matter.
Yes, that's great. It's the start of the system down here, and you'll notice that underneath that is dead material or material that are breaking down. So bits of stubble or old pasture or dead roots or growing roots, all coming into that area of organic matter, which, as I just said in the previous slide, is the food and the home for the biology. So if we can't look at just one aspect of it and think, well, we've got the environment there, but we don't have any food because they're not going to grow and repopulate, and we can't just put out foods and expect that the environment's going to be great and they're going to continue to grow. So from that, we can look at a bit of a food pyramid, which sort of simplifies it a little bit, in that if we're going to start at the bottom of the food pyramid, once we're talking about organic matter and breaking down that organic matter, the first thing that we're going to be looking at is algae and the microflora that are going to be providing food for bacteria. And bacteria are normally the one thing that we look at on our soil test results, on our biological soil test results, to give us an indication of the potential of that system. So if we have a system where we've got lots and lots of active bacteria, as we do here on Tim's place, Tim's biological soil test results for his control bay that we're going to look at today, have lots and lots of active bacteria. But if we look at the next stage up from that, which is protozoa and also nematodes, which are the middle of the system, we don't have hardly any of these. So these populations are very diminished, but we've got lots of active bacteria. So as we can sort of start to put together a picture, we're starting to realise that although the bacteria is fantastic, we're not actually converting that into available nutrients. So the nutrient conversion here is quite low because we're not turning over those bacteria. They're sitting there, they're populating, they're going nuts, but nobody's actually keeping them in control. And if we have a system that's very, very high in bacteria, we can often have potential for disease and disease pathogens because there's no control keeping them under wraps, so to speak. So as I said before, if we're just going to focus on fungi, which is very important, without focusing on the other ones underneath, without focusing on microflora and bacteria and protozoa and nematodes, then we're not going to get the full potential out of our fungi because they rely on those organisms in order to develop a good habitat for themselves. So we need to be looking at that not in isolation but in connection with everything else. And then microfauna, which is still able to be seen underneath a microscope. All these things have to be looked at underneath a microscope because they're too small for us to see. We sometimes see damage, as in, if we've got leaf or root feeding nematodes, we can sometimes see damage to our crops, so we might suspect that there's something there. We can draw vague conclusions to it, but without actually looking at it underneath a microscope, we can't get an adequate view of what's going on. So microfauna are still very, very small, and then the main things that we can see above the ground and in the soil, if we do soil sampling, is the macrofauna. So the big organisms that we can see, like slaters and other larger animals like spiders and ants and things like that. So thinking diversity, we need to think of the variety that's there and also the population numbers when we're trying to improve that. It used to be thought that if there was lots of macrofauna in the system, that was, like a good indicator for having all the other parts of the ecology there as well. And while I still think that's probably valid, we probably don't know, we probably haven't done enough testing in general in the biological farming community to be able to say definitely that if you have lots of macrofauna there, that the other things are there in the right populations and right ratios as well. So, macrofauna, because they can move, because they have legs and they're quite mobile compared to bacteria, which, can only move like micrograms of space, because they can move and they're quite mobile, do they come in and are they opportunistic and do they take advantage of certain times of the year and certain situations before harvest, which they could do. So maybe that's not the most accurate indicator. I think the best indicator is actually seeing that they're there underneath a microscope and seeing that they're active. And then it's almost a real-time thing. If you put a sample underneath a microscope, you're seeing it happening as it's happening. You know that they're there if you've taken a good enough representative sample of your paddock. Often when I'm talking to farmers or different groups about biological applications, people mistake biological foods for biological applications. How would we distinguish between the two? One's a food and one's got biology in it. So if we're to look at some of the common applications that go into biological farms, if we're to look at these, is compost a biological application or is it a food? It's both. It's both because it's a medium for the biology, so it's carrying the biology and it's also got the foods in it, so it's a perfect formula. Compost tea, food and biology, yep. So it's going to have foods in the compost tea when we brew it. We're going to add the right foods to sustain those populations. If we've made it incorrectly or we've added too much food or not enough food, then we're not going to have the right balance. Vermicast is going to be a food for the general biological populations, and it's going to carry worms and little worm babies, but it's not going to be in terms of a full suite of organisms. It's not going to have that full variety that we talked about earlier. Microbe extraction, which probably hasn't been done a lot around this area, which is not the full process of compost tea brewing, but actually extracting the organisms off a material, so you can have like a protozoa extraction off lucerne, so off like maybe a lucerne hay or off fresh cut lucerne. So it doesn't actually involve adding the foods and brewing them, it just involves taking them off the material and putting them into the water and then putting them out that way. So it's going to be a biological application in that we're putting out biology, and then we need to concentrate on what foods we're putting out, or what foods are in the paddock when we're actually putting them out. Inoculants is a bit contentious, because if they do have active biology, see the other thing with inoculants is that we can buy them, they're usually freeze dried, and then we have to wake them up. So it depends on the success of that waking them up, whether or not, how successful they may be in the paddock. So yeah, again, they are a biological application if they're containing biology. If they're just containing foods and dormant spores and they don't wake up fully, well, they might not actually have put out any biology in there, or very small amounts maybe. Seed treatments, depends. We can use compost tea and compost, well, not so much compost, but we can use compost tea in a liquid form as a seed treatment. We can put out compost as a broadcast application when we're putting out our seed, or if you're in a row crop system, you can put it out in your row crops. So it can be used as a food and a biological application. Seaweed, this is where it starts to get into the food section. So seaweed has a lot of micronutrients and enzymes and things which are great stimulants of the biological system, but they're not necessarily carrying any biology. So unless they're used in a compost tea and they're being put out with actual microorganisms, then they're not a biological application as such. They're just a food that you're putting out there, or a stimulant. Same with humates and humic acids and fulvic acids. How many people have heard of humic acid and fulvic acid? Yeah. So commonly commercially available now, and can be used to buffer things like urea applications, which are highly salty, which we want to minimise those effects when we're putting them out into the paddock. So they're also a food, and we have used some in the compost tea brew, but not necessarily, if you're just putting them out by themselves, they're not necessarily a carrier of biological organisms. And the same with fish emulsion, as same as seaweed, it goes out as a food and doesn't necessarily have the biology in it. In terms of trying to source something really valuable, manures I tend to think that they're less valuable than a compost or a compost tea, because they generally don't have that biological population in them that's going to help improve soil. So if you're just looking at it from a nutrient perspective, that's okay if that's all you need. So in terms of applying the biology, I'll probably add another note in here. So if any of you have got pens or pencils with you, you can actually change this one. The compost, we were putting it out at 2 to 4 tonne to the hectare, but I've recently received some information from people who've put it out at, say, maybe even half a tonne or a tonne to the hectare, and that's still giving them good results. So don't feel that if you can't put out 2 tonne to the hectare that you shouldn't put it out. If you've got access to any compost and you can put it out at any rate, it's going to be doing more for you than not putting it out, if it's good quality, if it has all the microorganisms in there. And because in Australia, I think they've only recently developed a standard for compost in Australia, but up until then, compost can be referred to as sort of mulches. So just like it's called composted pine bark, but really it's just pine bark that's been wetted up and sort of let to rot in a pile and then turned with a front end loader, and that's not compost. So if again, composted cow manure, you'll see that in advertisements. If it hasn't been through an aerated cycle, so it hasn't had a thermal cycle coupled with aeration and good monitoring of the biology and good monitoring of the nutrients, then it's probably not worth getting. What I've found in my own situation on the farm over here is that the best diversity going into the pile initially gives the best diversity at the end. So if you just have chicken manure, green material, say you're doing it on a large scale like the composters down south are doing, and getting green material from the cities, so it can be anything from grass clippings, local green waste and veggie scraps, and say straw, say you're going to use like a cereal like wheat or barley straw, then it's only really 3 different sort of types of ingredients. Maybe you get more variety in the green materials, but it doesn't tend to produce enough resistant carbons that will provide a lot of fungi. So you can tend to get a really good compost, like a balanced mix, but it doesn't tend to be very fungal dominant, and this is going into composting now, a bit different to what we're talking about today. But if we're trying to improve that fungi, we want to have all the other things in our pile as well as the fungi. And fungi takes a lot longer to develop in a compost pile, and especially if it's being turned and manufactured in large windrows, it tends to be harder to get that fungi into the pile and keep it in the pile because it doesn't like to be turned as regularly as the bacterial stuff does. So in terms of that, we'll talk about it a little bit when we talk about the compost tea. But I think if you're going to try and increase fungi in your system, then you probably want to do smaller amounts of composting, better quality, and do compost teas rather than trying to get large amounts of, say, average quality compost. Well, if you can make a good fungal pile in 2 months, like over a 2-month period, then making a compost tea only takes like 2 days. If you think about, say, barley straw or wheat straw, it's quite an easily digested carbon product, so it's quite thin, you can scrunch it with your hand, it's not very hard and tough. The carbon that's in it is sort of small chain rather than long chain carbons. So the fungi prefer the more resistant stuff, like if you go out and see a tree fallen over in a forest and fungi growing on that, that's the stuff that they love, is the stuff that's going to hang around for a really, really long period of time, that they can digest over a long period of time. If we don't have that in our pile, then obviously we're not going to be providing the food for them, so they're not going to they're not going to develop in the pile. We'll get smaller amounts of sort of mushroom style fungi happening from spores that have germinated in the pile really quickly. But in terms of whether they stay around long term, if their food's not there, if they don't have really chunky woody bits in it, then it's probably not going to end up going the full way to being, a very fungally dominant pile. Compost, I would say, if you can get hold of it, and if you can put it out at half a tonne or a tonne to the hectare, still going to be beneficial. Don't have to aim for the higher amounts.
So how often are you looking at applying?
Well, depends on your budget. If you can put it out once a season and follow up with compost tea, and do maybe 2 or 3 compost tea applications during the season, that's what we're doing at the moment. I've got a compost area that I've put out compost, and I'm going to be following up with compost tea applications and tracking those results, as opposed to areas where I've just done compost tea. But really, if you're monitoring your system and you're taking soil samples regularly and monitoring those populations going up or down or staying stable, then you'll be able to tell whether or not you need to add more of a certain organism to get the system to be stable. Does that make sense? Yep. Compost tea that we're doing today, we normally put out between 30 to 50 litres. Sometimes I go up as high as 75 litres or even 100 litres if it's average quality. We're hoping that the stuff today is going to be great quality. It's been brewing for probably about 40 hours, maybe a bit longer, 48 hours. Saturday night, Monday. No, it's probably been brewing longer than that, 48, maybe 50 hours actually. And I haven't been able to do a microscope sample of this, but I will do one when I get home tonight and let you all know what was in there. And because it's colder at this time of year, because the temperatures are colder, it's going to take longer for those organisms to develop. So even a couple of weeks ago when we did the one for Kerrang, it was still a little bit warmer. And so brewing doesn't need to go for as long a period. You can stop it a bit earlier and still have good development. So again, compost tea, you want to test that underneath a microscope or send it away for a sample, same as you would with compost. So just going back to the compost, if you're going to buy it, make sure that whoever you buy it from has done some sort of testing on it and some sort of regular testing, not just like a one-off test that they did on their first batch and then just keep whacking that one out every time, because each batch is going to be different. So if you're going to spend a fair amount of money on it, then you want to make sure it's good. And to do a compost sample, I think is worth it, just to make sure that you're going to put out something good. So inoculants, if we're going to apply those, it's probably going to be something specific like a VAM inoculant or a rhizobium inoculant. So it's not going to have the same diversity that it would if we put out, say, a compost tea seed treatment. If we've got all the organisms in the compost tea, then we're going to get a lot better cost effectiveness out of our compost tea and all those organisms, as opposed to buying just one isolated strand, or even a commercial product that maybe have 3 or 4 strands of one organism type. Or we do ours in a little concrete mixer with bags, which is probably more labour intensive than some people have the little drip meter thing on their auger as they're augering it up. So yeah, it's just enough to coat the seed lightly, but not enough to make it sludgy or wet, and not that it's going to affect its germination. So just enough to coat it on the outside, because some of the fungal spores and the fungal populations, they have to come into contact with an active root system. So as the seed germinates, you want to have those spores right next to it, so that as the roots come out and start to develop, it's going to be right in contact. You don't want to be putting it out on the soil surface and then be sowing like this far down. So that's why the seed treatment works sort of better than just putting it out as a surface application. You're better putting it out as a surface application once it's popped out of the ground. Okay, so we talked about seed treatments and the extracts. So particularly protozoa extracts are popular. We've got a little amoeba there. So if we're trying to encourage protozoa into our systems, then we might do an extract rather than a compost tea, and put that out as a foliar spray or as a seed treatment, but probably more as a foliar spray, because it's going to require bacteria and things on the leaf surface to get it going. Going back to what we said in the first slide, if we're going to be brewing compost and compost teas, we need to be thinking about keeping them in the system. So the main thing we talked about was the food and the home. So if you only remember 2 things from today, think about what sort of food is going to be out there in your paddocks, and what sort of home are they going to go into. If we're talking about increasing our bacterial populations, which for most of us who have got rundown systems, everything's going to be low. So usually starting with either a balanced compost or a balanced compost tea. When I say balanced, I mean has as much of everything in it as we can get. Maybe not super duper fungal, but we'll have a good amount of each in the pile or in the compost tea. If we're trying to encourage those, we want to have fast sugars and green material. So when we say fast sugars, we might be able to put out something like a light molasses spray just prior to putting it out there, so that they've got some energy to keep them going. I always tend to think about it in terms of humans and low GI and high GI foods. So if we want low GI foods and we want to sustain our energy over a very long period, we want something that's going to slow release and keep us going for a long period of time, which is what the fungi wants, the long period of time to develop, and those resistant carbons. So if we've got stubble in the system and we've got a variety of different stubbles, maybe over a couple of different years, we're going to have different rates of breakdown. So we might have stuff that's already half broken down and stuff that's quite fresh. So it's quite a good environment to put them out into. If we're going to put them out onto a chemical fallow or a mechanical fallow, their foods aren't going to be there and their home isn't going to be there. So probably not advisable until you get something growing in there to put out a compost or a compost tea, just because it's going to be a waste if they don't survive, or if only a small portion of them survive. It's still, unless you're putting it out there for free and you're just doing it for the love of it, then you want to be putting it out there at a time that it's going to be at its best. So porosity is going to be probably Tim's major issue here, and because he's very, very heavy clay soils, if we go look at further down into the soil profile and we were to do, say, some 1 metre soil cores, it'd be really interesting to see how, whether or not there's a compaction layer there, and if so, how compacted it is. and how much oxygen is diffusing through that soil, because the main thing that's going to regulate those populations is oxygen content around those soil particles and moisture content. So it doesn't necessarily have to be wet, but soil will hold on to moisture around those surfaces of the particles. So if we were to take a soil sample and break it up, and we can actually break it up into those smaller particles as part of that core, then usually we've got some decent porosity in terms of that the oxygen and water will be able to diffuse through there in the right scenarios, i.e. when it rains. If we go to break open our soil core and it's like concrete and we can't break it open, there's no air getting through there whatsoever, then that might be a bit of an indication that your biology aren't going to get down that far, because they can't penetrate stuff that's like concrete, and they're not going to live there anyway because there's not going to be any oxygen and probably very little moisture. So just thinking about that when we put them out. So along the outside of the particles is where we're going to get most of our macropores, and then the smaller, finer particles, which are going to, or gaps if you like, which are going to be in between those bigger particles, is where we're going to find a lot of the bacteria and protozoa and things, because they love to live in those little, if you think of it like streets, like little cul-de-sacs and things that are going to be in those tiny, tiny little spaces. That's where they like to live, and the fungi are going to be going along the superhighways between the bigger gaps. So where old roots might have grown and died, where we can actually see that there's visible spots. And sometimes if you have good fungi in a paddock and you take up a soil sample and you break it open, you can actually see where a root hair has maybe died, and you can see colonisation of fungi that has maybe digested that and then moving along to different spots to find more food.